What are the molecular techniques for cloning a gene? Well, after isolating and purifying the DNA containing the desired gene, use restriction enzymes, which can cut DNA at specific sites, to separate the desired gene from the rest of the DNA. Use the same restriction enzymes to open up plasmids, circular pieces of DNA, and insert the desired gene. The complementary ends will match and stick together. We then transform bacteria to incorporate the plasmids containing the desired gene. These bacteria begin to grow into large colonies that can be seen with the naked eye. At this point, we can identify the bacteria that have successfully incorporated the desired gene. In many cases, antibiotic resistance is included in the plasmid. Taking advantage of this resistance, we can put the bacteria on a plate with antibiotics. The bacteria that survive have successfully incorporated the plasmid. However, the bacteria may have incorporated part of the plasmid without incorporating the desired gene. Sometimes, the bacteria with the desired gene will turn a different color from the bacteria without the desired gene. To ensure that the bacteria do contain the gene, we can check the bacterial colonies' color. Then we can isolate these bacterial colonies, which now consist of many bacteria, all potentially containing the desired gene. Cloned genes have many uses in research and medicine.
Dolly is without question a normal, healthy, genetic marvel. She's an identical copy of an older sheep.
People expected that maybe at some time it would happen, but maybe we've been a little bit quicker than they expected.
Dr. Ian Wilmot and his team of Scottish researchers began with a 6-year-old sheep. They took a single cell from her mammary gland, including its nucleus, the command center containing all the genes, the DNA, that determine the sheep's individual physical characteristics, eyes, nose, hair. Then they took an egg from a second sheep, removed its nucleus, and replaced it with a cell from that first sheep. The egg and its new nucleus then fused together, turning into an embryo, but with all the genetic characteristics of the first sheep, and none of the second. In effect, a carbon copy, an exact duplicate of the first. All that was left was to put this developing embryo into a third sheep, a host sheep, which carried it to term.
The key to this experiment was to induce the genetic information of a very specialized cell from an adult, in this case a mammary gland cell, to go back through development to produce an entire new animal.
What has caused such a shudder in the worldwide scientific community is that so many scientists doubted it could ever be done. They had tried to clone frogs and then mice, and without success. This latest research on sheep did not rely on any revolutionary new technology. In fact, many in vitro fertilization clinics use the same equipment. But it's clear this cloning technique still requires some refinement.
We've succeeded in setting up a system which will work sometimes, but we don't remotely begin to understand why. It just happens from time to time.
Wilmot says it took nearly 300 tries to create this one lamb. John Mackenzie, ABC News, New York.